Okay. So hi guys, welcome to week 5 practice session for this course, that is cell biology. Let me just share my screen and we can briefly, go through my introduction and then we'll begin the discussion for today. Yeah. So I hope my screen is visible to everybody. Uh, yeah, so the volume is fine. Hmm. Hi guys. Those who have not attended any of the previous practice sessions that have been conducted by me, for them, let me just briefly introduce myself. I am Arveen Kaur. I am a PhD scholar at the IIT Kanpur, and currently I am also a teaching assistant for this course that you guys have been attending, that is cell biology, right? So how we'll go through today's lecture or today's practice session rather is that in the first half of it, we will discuss the previous year's assignment pertaining to the week 5 lectures that were delivered by Professor Shikha Laloria, right? So because your assessment for week 5 is going on right now, we cannot discuss the current assignment, but there might be some concepts or some hints and clues that you might be able to pick from the previous year's assignment, which might align with your questions or how to solve your questions this year as well, right? Apart from that, even if the questions are very different, and if you have indeed, registered for examinations and all, it will help you in the long run because in those examinations you can actually, once revise the previous year questions as well. So you will have a larger database of questions to cover from. So having said that, we will begin with the previous year's assignment, right? Just a moment. Yeah. So the first question in last year's assignment being the differential localization RAN GTP and RAN GTP in in cell compartments is important because A, it helps in the formation of nuclear pore complex. B, it provides directionality to nuclear import. C, it helps in recognition of the NLS or nuclear localization signal. Or D, that is none of the above. So, which of these do you think is like the role of having two kinds of RAN, phenotypes? One is the RAN GDP and one is the RAN GTP, right? So there are two kinds of different, you know, presentations of these proteins or two different kinds of complexes of these proteins. What is its primary function? So go through the question once. Yeah. And we can discuss its answer then. So the answer for this particular question is that this difference or this differential localization of these two complexes of RAN provides directionality to the nuclear import, right? So this is something that we have gone through while ma'am was discussing, and her TA also while she was discussing different kinds of nuclear localization and meiosis in those lectures. We have studied about RAN, right? RAN is a protein which basically carries some cargo into and out of the nucleus, right? So there are two kinds of complexes that it will be involved in. One is the RAN GDP complex, right? The other is the RAN GTP complex. RAN GDP complex is generally you sorry. Uh, Yeah, so RAN GTP complex will be involved in taking the cargo inside, right? Uh, uh, no, so there is some problem with this representation actually. RAN GDP will buy into whatever cargo has to be taken inside the nucleus, right? It will there interact with the GF, which will convert this GDP subunit to GTP, wherein the binding for this cargo with the important will kind of reduce, and this will lead to dissociation of this cargo inside the nucleus, wherein then this important RAN GTP complex will be bound and it will go outside, right? And once it goes outside again, there the it will be RAN GTP over here will be converted to RAN GTP, and the important will be left free, uh, so that it can bind to a cargo that has this nuclear localization subunit, and it can actually come back to the nucleus, right? So in writing also I have mentioned this. So this is a representation that has been picked up from the book that we've been talking about in a few of our previous lectures also. This is molecular cell biology, or as most of you might know it by the name of Lodish, right? So, what happens in this RAN GTP complex is that in the cytoplasm, the free important binds to the nuclear localization signal of a cargo protein, forming an important cargo complex, right? The important cargo complex, as we see over here, diffuses through the nucleopore complex by transiently interacting with the Fg nucleoporins. So these nucleoporins are rich in Fg, right? And by transiently interacting with this, it can diffuse into the nucleus. In the nucleoplasm, the RAN GTP binds to the important, right? And this causes a conformational change that decreases the importance affinity for the NLS, right? When GTP is going to interact with important, its affinity for this particular cargo will decrease, and the cargo will be left inside the nucleus only, right? To support another cycle of import, the important RAN GTP complex is transported back into the cytoplasm, as we saw, and a GTPase activating protein or a gap associated with the cytoplasmic filaments of the nucleopore complex stimulates RAN to hydrolyze the bound GTP, right? If this hydrolysis of this bound GTP occurs, it is also going to drive this other cycle of import, because it is going to produce some kind of energy, right? This generates a conformational change that causes RAN to dissociate from the nuclear transport receptor, which can then initiate another round of import, right? Then the RAN GDP is returned to nucleoplasm, where a guanine nucleotide exchange factor causes release of GTP and a rebinding of the GTP, right? Okay. So coming back to our next question, which is question number 2, it says, which of the following option correctly describe an event occurring in the prophase 1 of meiosis, right? So go back, think about meiosis, think about this particular phase in meiosis, and here are the options for you guys. Option A is pairing of homologous chromosomes and crossing over. B is alignment of homologous chromosome at the cell equator. C is duplication of DNA, and D is segregation of homologous chromosomes. So which of these do you think will correctly describe the events that occur in prophase 1 of meiosis? So for people who have gone through this week's lectures, will be very well versed with this, right? So prophase 1 is a very long, relatively long phase, right? That occurs in meiosis. And the correct answer for this one is pairing of homologous chromosomes and crossing over, right? So this is actually one of the key features that distinguishes meiosis also from mitosis, and right? That over here, what happens, the homologous chromosomes, they are not really separated or segregated as the cell divides. At least in meiosis 1, first, they stick together, right? So some kind of pairing occurs between these homologous chromosomes. And while they are like bound together, they also undergo a process called crossing over, right? This is important for recombination to occur. So this I hope everybody has gone through. I am just going to zoom in this figure that I have kept. Uh, yeah. What happens in meiosis is that in the meiotic S phase, there is again a division or the duplication of DNA that goes on, right? And we over here see, suppose a gamete which has a paternal homologue and a maternal homologue. And over here, we are considering just two chromosomes, right? So that, or just one chromosome rather, so that it is easier for us to understand, right? In the S phase, like in the mitotic S phase, the replication of DNA occurs, and now they have kind of formed, doubled in the DNA, right? What happens during mitosis is that these two chromatids will go towards different poles, and they will segregate into two daughter cells, and then again a diploid nature will be maintained. But over here, this diploid cell became 4n when DNA replication did occur. Right after that, during meiosis 1, what happened was that there was pairing and crossing over of duplicated homologues, which is what we have talked about over here, right? That there is a pairing of homologous chromosome that occurs, and crossing over also takes place, right? After this pairing had occurred, the homolog pairs line up on the spindle, right? So, like there was that metaphase plate that occurred in mitosis. There is a metaphase, in the center orientation of these homologs occurs in meiosis one also, the metaphase one of meiosis one. Now, at anaphase one of meiosis one, what happens is segregation of homologs occurs, right? So instead of sister chromatids over here separating, there is segregation of the entire chromosomes that occurs, right? Which is how it is different from mitosis. Then after that, cell wall will or cell membrane will form around these separated ones. After that, meiosis two will occur, which is more common like mitosis, except for the fact that at the end of this meiosis two, since the beginning is a diploid number, at the beginning of mitosis, we have a tetraploid. So the result is a diploid, right? Over here, since the beginning, substrate is a diploid cell, that is why the end result is haploid daughter cells, right? So I hope you were already aware of this, right? But we have revised this once again. Now we'll come to question number 3. Pick the correct order of different stages in meiotic prophase. Right. So the prophase that we talked about, we just talked about it as prophase one, going to metaphase one, then anaphase one, telophase one, cytokinesis, right? But uh, among themselves, like among the meiotic prophase itself, it is divided into multiple subphases, right? So these are the names of those subphases. We have to pick the correct order of their existence, right? So chronologically, like which phase comes before which other phase, right? So option A over here is leptotene, zygotene, pachytene, diplotene, and diakinesis. Option B is leptotene, pachytene, zygotene, diplotene, and diakinesis. Option C is leptotene, zygotene, diplotene, pachytene, and diakinesis. Or option D, which is none of the above. So I will give you a moment to recall this, uh, and then we can discuss the answer. Just a moment. So we are coming back to this question. The answer for this particular question is the first one. That is, this is actually the correct chronological order in which the meiotic prophase substages actually occur. So the order goes like leptotene, what happens in leptotene? In leptotene, the nuclear envelope has not yet dissolved, right? The duplicated DNA has still not condensed completely, so it looks like it still has that thread-like appearance, right? More or less. So it looks like that chromatin thread. In zygotine, what happens is that the DNA has become a little more condensed. Sorry, along with that, what has begun is the formation of a special kind of complex that is a synaptonymal complex, right, between the homologous chromosomes. Then after this, what occurs is a packetine. So packetine is one of the most important phases of prophase 1, right? Meiotic prophase 1. Because what happens in packetine, the most characteristic feature of meiosis, right? That is a crossing over actually occurs. Crossing over and recombination occurs in this particular stage, that is packetine, right? Then in diplotene, what happens is that the synaptonymal complex that had formed between the homologous chromosomes, now it starts to kind of separate. And in diakinesis, this synaptonymal complex has completely separated, right? The chromosomes are still bound to one another at specific points where the crossover occurred. These points are known as chiasma, right? Or chiasmata if there are multiple such points. Uh, and after this, the onset of the metaphase 1 begins, right? Then comes question number 4, which is synaptonymal complex disassembly takes place in which of these stages? So this is something I've just discussed, but let us go through the options. Is it leptotene or diplotene or packetine or zygotine? So while I was discussing the last question, I went through this as well. So hopefully you were listening and you do know the answer for this one, right? So the answer over here is diplodine, right? So as I explained, what happens is that, for example, over here the prophase begins, right? Before this prophase was interphase, right? Probably G2 phase of interphase was existing, right? During the G2 phase of interphase, as you can see in this particular region, right? What can you see that the chromatids look very distinct, right? You could recognize that this is one chromatid, this is one other chromatid, this is another chromatid, right? But as the prophase will begin, since the condensation will start occurring, now after a while, you will not be able to recognize in the first stage of this prophase 1, that is leptotene, you will not be able to distinguish properly, like in this out of this which one is chromatid 1 and which one is chromatid 2, right? Over here we can still see that some portion of chromatid 3 and 4 are like dissociated yet, so we can still make out, but as we progress through leptotene, that will be harder for us to make it out, right? And that is what occurs majorly in this first phase of prophase 1, that is leptotene. But in zygotene, that is the phase that follows leptotene, what happens is synaptonymal complex assembly begins, right? What will this synaptonymal complex do? It will bring the two homologous chromosomes, the paternal chromosome and the maternal chromosome. It will bring it together, right? So this is where the assembly of synaptonymal complex will begin. After that comes this most significant stage that we talked about, packetine, wherein crossing over and recombination of between the non-sister chromatids, probably chromatid 2 and chromatid 3 of these homologous chromosomes will occur, but it could also be chromatid 1 with 3 or chromatid 1 with 4 or chromatid 2 with 3 and or 2 with 4. Okay, but the crossing over will not occur between chromatid 1 and 2 and between chromatid 3 and 4 because they are sister chromatids. They are not non-sister chromatids, right? We have discussed this previously in, I think, week 2 or 3 lecture, right? Uh, then after packetine, the stage that follows is diplotin, right? And then comes diakinesis. So in diplotin, that is the stage where disassembly of the synaptonemal complex will start, right? And by the end of diakinesis, it will totally have disassembled, right? So yes, diplotin is the answer over here. Okay. Okay. So, question number 5 is non-disjunction due to improper segregation chromosomes may lead to which one of these? So, is it aneuploidy or Down syndrome or progeria syndrome or none of the above? So, those who are actually trying to solve questions as we go along, let me just give you a little hint that the answers or the options are not entirely correct, right? So, that is the only hint I would like to give, and we will definitely discuss the answers. But if you are trying to solve it on your own, that was the hint from my end, right? Okay. So for this, I hope you remember what non-disjunction is, right? And what these terms signify: aneuploidy, Down syndrome, progeria syndrome, etc. Right? I hope you remember that. Yeah, somebody raised their hand. I'm sorry. Huh. I cannot really see your name at this moment. Can you just turn on your mic if you wanted to speak something? Okay. Anyway. So yeah. So as I give you a little hint that the options given are not entirely correct, right, for this question. So why did I say that? Because two of the options could be considered right, right? So what is non-disjunction? For example, for the first thing you need to know what is non-disjunction, right? So, what does these words like if we break them down, what do they signify? So, non means something that is not happening. Disjunction, right? So, something that has to be broken from the junction is not happening, right? That is what is known as non-disjunction. So over here, why are we saying that there could be multiple reasons actually due to which non-disjunctions could occur, right? But these are usually taken care of by the regulatory mechanism or regulatory, uh, uh, this force that is implied on by the cell. Sorry. But sometimes they get overlooked, right? Due to which some conditions may arise or some improper segregations of chromosome will occur, right? So what could be non-disjunction is, for example, this is a set of chromosome, right? So it ideally had to be separate, joined to the spindle or the microtubules so that after it got segregated, right, one chromatid went over here, one chromatid went over here, right, and a cell formed around them, right, cell wall formed around them. So this would be a proper segregation, right? Sometimes what could happen is that due to some error, maybe in the spindle attachment or maybe in the centrosomal pulling mechanism, maybe both the microtubules, kinetochores, attached to the same end of the pole, right? In that case, what will happen is that two of the cystochromatids will get pulled to one end and there will be nothing on the other end, right? And a cell wall will form around this and another cell membrane will form around this. So this one will not have any of this chromosome, right? And this one has two copies of this chromosome. Both these conditions are hazardous, right? So this is also not good for us. Having a double copy of anything does not mean that it will benefit us, because too much of something could even be toxic for us, right? So a lot of neurodegenerative disorders, for example, are you know are the correct instances for this thing only, because some kind of a repeat expansion occurred. For example, in Huntington's disease, this happens that there is CAG repeats that are present in our genome. And normally these CAG repeats are ranging from around 25 to 35. That is tolerable, right? And that is actually the normal condition also, because they do have some function to play, these CAG repeats, right, in our genome. But sometimes what happens, if the number of repeats increases, and especially if it goes beyond 40, right? Sometimes it might go even till 50, 60, 70. What will happen? You will face a kind of disorder, right? What is this disorder? Huntington's disease. And why are you facing it? Because too much of the product that was being produced by that CAG repeats is toxic to you, right? So too much of something is also harmful for us, and too little of something is also harmful for us, because now we don't at all have any product that was that these chromosomes were going to form, right? So if we don't have that product, maybe it was important for our survival, and maybe we will not be able to survive. And maybe we did survive, and we were born into this world. But a lot of normal things that are very normal for others, we might not be able to do, right? So this is what non-disjunction is. All right. So non-disjunction could lead to improper segregation of chromosomes, right? Now, if this happens, then either of these conditions could occur, right? It could either be aneuploidy or Down syndrome. So, why do we say that? Because what is aneuploidy? So, for this, I have kept a definition from Wikipedia. That is why I had mentioned that link. Uh, oh, the link was for that picture. Yeah. So aneuploidy is an abnormality in the number of chromosomes in a cell which is due to loss or duplication, right? In humans, for example, aneuploidy would be any number of chromosomes other than the usual 46. So even if it is 45 or 47 or maybe 38 or 48, whatever the number is, if it is not 46, it will be an aneuploidy, right? So loss of chromosome or gain of chromosome or set of chromosomes, both of them come under this condition, which is aneuploidy, right? So if non-disjunction does not happen, so aneuploidy could occur, right? And the second option that I have highlighted here is Down syndrome, right? So what is Down syndrome? It is basically the presence of three 20, chromosome number 21, three copies of chromosome number 21, right? So in some cases, it might happen that the non-disjunction of chromosome or segregation of chromosome did not occur properly, right? So one of the cells that is now formed has three copies of chromosome 21, and the other one only has one copy of chromosome 21, right? Ideal case scenario, what would it have been? That both of these cells have two copies each of chromosome number 21. But sometimes it might happen that one cell has three copies of chromosome 21, the other one has one copy of chromosome 21, right? This trisomy, or three copies of 21 being in a chromosome, it might not be very lethal to the developing embryo or developing fetus, right? So that is why It is relatively common as a genetic condition that there are a considerable number of babies that are born with Down syndrome, right? Because it might not, pose any developmental defect. But once they are actually born, they do have abnormal, thinking capabilities, abnormal brain size, then their mental health is a little retarded. So these kinds of postpartum development, there might be some problems, right? But it does occur. So a trisomy of 21 could also be a result of non-disjunction and improper segregation of chromosomes, right? So that is why either of these answers would have been accepted. Progeria syndrome is another, progressive rare genetic disorder, but it is not due to improper segregation of chromosomes, right? And you guys might not even have heard about this one. Down syndrome, on the other hand, I would assume that at least, if not everybody, at least 70-80% of you might have heard about this, right? Because it is, like I said, it is relatively, common genetic disorder, right? And I told you why it might be a relatively commonly seen genetic disorder, right? Because during the fetal development, it might not pose any kind of harm, right? So over here again, there is a representation of non-disjunction like the one that I had drawn earlier. So that was a very pathetic drawing, but this is a more sophisticated representation of what we were trying to say, right? So at metaphase 1, even sometimes what might happen is that there is normal attachment of microtubules, right? Even after that, maybe due to some reason, uh, the attachment of this particular side of microtubule was stronger, or the pulling force was stronger than this particular centriole on this particular pole, right? Due to which there could be multiple factors, due to which the chromosome or the chromatids are pulled to one center altogether, right? So thus these non-disjunction conditions could arise. Okay, so I hope everybody's clear with that. Uh, okay, so question number 6 is, during homologous recombination, at the point at which the chromatids from a pair of homologous chromosomes are physically attached to each other is called as A chiasma, B centromere, C telomere, or D kinetochore. So think about this question. Multiple of the options are actually points of attachments, but over here, what has been asked to you is that which of them, which point of attachment is, occurs during homologous recombination. Okay, so I think we can discuss the answer. Just a moment. Yeah. So the answer for this particular question is chiasma, right? So during the earlier questions, I did discuss chiasma, chiasmata, right? So what is this? It is basically the point at which the chromatids form a pair of homologous chromosomes, from a pair of homologous chromosomes are physically attached to each other, right? These usually signify the points wherein the crossovers have successfully occurred, right? And these chiasma will remain, this is how the homologous chromosomes will remain bound to one another. This is one of the features, right? So over here, for example, we have a representation of the chiasma. First, let us go through this definition, right? Chiasma is an X-shaped connection between paired homologous chromosomes during meiosis. It represents a site of chromosomal crossing over a form of genetic recombination, right? So, for example, we say that this red one is one chromosome, one homologous chromosome, that is paternal chromosome, and the gray one is its other homologue, that is a maternal chromosome, right? The two arms in the gray one are sister chromatids among themselves, and the two arms in the red one are sister chromatids among themselves, right? These two arms, like one red and one gray arm, are non-sister chromatids, right? In the middle or towards one end, it could be either way, there will be a place wherein the two sister chromatids are bound together, right? This binding point is known as a centromere, right? Now, this complex, during crossing over or after crossing over, it forms this kind of a structure, right? It is mentioned an X-shaped connection, right? This kind of a structure is formed, and where they are joined together, the two non-sister chromatids, where they are joined together, it is known as a chiasma, right? So one of the key features to remember how chiasma is different from a centromere is that centromere is a connection or joining spot between of two sister chromatids, right? While chiasma, this was centromere. If I said something else, centromere is a point wherein two sister chromatids join together, right? Are joined together. Whereas chiasma is a point wherein two non-sister chromatids are joined together, right? So I hope that will help you remember this. Okay.
Ma'am, can you just repeat what you said?
Okay. So what I said was that one of the key differences between chiasma and centromere is that in the case of centromere, it is a point of attachment of two sister chromatids among themselves, right? It could be the red sister chromatids attached together at this point, which is known as centromere, or the gray sister chromatids attached together at this point, which is also known as centromere. On the other hand, chiasma is a point of attachment between non-sister chromatids, right? So sometimes these differences are asked. So for that, I mentioned this thing. I hope it is clear. Okay. So moving on to question number 7. Question number 7 says, in budding yeast, the meiotic commitment initiator IME1 acts as which one of these? Does it act as a phosphatase or a protease or a transcription factor or adapter protein or a kinase? So there are 5 options that have been given to you for this particular question. Go through the options, try to remember that we have actually studied this during this week's lectures. Mm, so I'll give you a moment to recall and then we can discuss the answer. You remember when this budding yeast was being discussed, it was also spoken about that during some different kind of nutrient conditions, they will start this meiosis, right? So there will be some factors that will be involved for that, and one of them will be IME1. So that is the part you have to recall. Now, IME1 in that case acts as which one of these, right? Is it a phosphatase or a protease or a transcription factor or an adapter protein?
Transcription factor, ma'am.
Yeah, so that is actually correct. So, IME1 helps in commitment to meiosis by acting as a transcription factor, right? So, like we had discussed, so I would request you to omit the unnecessary, I have not modified this particular representation that I have picked up from this paper by Wiedberg et al. From PLOS Genetics, right? Over here, we will just look at what is necessary for us, right? So what happens during a nutrient-rich condition? This IME1 promoter is like inhibited, right? You can see this that there are multiple pathways due to which the IME1 promoter is inhibited when there is a nutrient-rich condition, right? So what happens when this promoter is inhibited? There is basically no meiosis that can occur, and no sporulation also occurs, right? Because the spores for sporulation is basically the formation and dispersion of spores. So spores will only be formed when meiosis will occur, right? Spores are haploid uh, in nature, whereas the normal budding yeast is diploid, right? So if meiosis will not occur, sporulation will also not occur. But when sporulation does occur, right, we have from our lecture, we know that when there is nutrient deficient condition, sporulation can occur, right? So whatever pathways were actually inhibiting IME1 at previously during the nutrient rich condition, now these pathways are not working that way, right? So IME promoter gets activated, sorry. So the IME1 promoter gets activated and it acts as a transcription factor, right? And it will transcribe the necessary proteins that are transcribe the necessary genes, which will lead to the translation of the necessary proteins that are responsible for the commitment or the onset of meiosis, and thus also onset of sporulation, right? So this is how IME1, will actually act as a transcription factor for meiotic commitment, right? Now, apart from this fact that we had discussed in during our normal lectures also, there was another thing that I actually wanted to, take your notice to. So I am considering, or in an unassuming way, let us consider that not everybody is from a cell biology or a biotechnology background, right? So for somebody who is not from cell biology, biotechnology, zoology kind of a background, somebody might have a little problem in understanding that why at all, when we have nutrients, we are not, producing gametes, and when we don't have nutrients in a nutrient deficient conditions, we are actually producing gametes, right? So that is not what actually happens in higher organisms, right? For example, if we talk about mammals, such as dogs, cats, or even humans, right? That is not the case that is going to happen, right? So only if you have enough nutrition for yourself and for the people that are around you, will you actually be able to provide more nutrition to your offspring also, right? So you will only reproduce probably when you have enough nutrition, right? Now, that is not always the case. In lower organisms, uh, the sexual reproduction is a mode of survival, right? So, why would anybody reproduce sexually when they can actually perform asexual reproduction also? And asexual one might be very cost effective, right? That there is with this one organism, diploid organism, it can just form a bud, and that bud can grow up into another diploid organism. So, this is not costing much in terms of energy, in terms of nutrients required, or in terms of the general supply of proteins within that organism. It might not be that energy intensive, right? So that is why usually what happens is that the yeast, which can both reproduce asexually and sexually, if the conditions are favorable, it will continue to reproduce asexually, right? But sometimes what will happen now, the conditions are not favorable anymore, right? The nutrients that it required for this asexual reproduction are now not available, right? So if it doesn't reproduce and something happens to this existing yeast also, then probably it will die, and it could happen that yeast in general die, right? So that is not what happens. Under this very stressful condition or nutrient deficient condition, what happens is that the yeast will form a covering around it, right? And within this covering, what will happen is inside this, this diploid yeast will undergo, yeah, Netruvati, yes.
Excuse me. Yes, yes.
Yeah, yeah.
So I could not able to view your screen. So I'm trying to log in from the other mobile by name Balasubramaniam. So can you let
Okay.
me in?
Yeah, yeah, yeah. Did you try, like, try, please, try again? I'll let you in. So let me stop sharing my screen so that I can actually view who is joining. Uh, Dhana Shekhar, you know, yeah, okay. So I have let you in, okay? So I hope now my screen will be visible. Currently, I'm not sharing my screen, but
Okay.
I'm just going to.
Thank you, ma'am.
Welcome. So I hope it is visible now.
Yes, yes. Thank you, ma'am.
Okay. Yeah. So what I was saying was, yeah, what I was saying was that during a favorable condition, yeast is going to continue to, reproduce asexually. But under nutrient deficient conditions, when it actually comes on to the life of that particular yeast, it and one of the basic survival instincts for all organisms is to reproduce, right? Survival instincts in the case that they want their genetic material to exist, keep existing, right? So that is why now this asexual reproduction might not be favorable anymore under nutrient deficient conditions, and the kind of environment outside might be harmful for this particular yeast. So what it will do, it will form a kind of barrier around it, right? This barrier is a very, very strong barrier, and it is resistant to most kind of stresses that are there in nature, right? So within this barrier, what it will do now, this diploid yeast will undergo a sexual reproduction, and through sexual reproduction, it will, form four different spores, right? Haploid spores. So that is how sometimes during nutrient deficient conditions for its survival, this sexual reproduction is taken up by the budding yeast, right? And after the conditions are favorable again, this outer coat or outer covering that was actually protecting this particular yeast, it will be broken down and these spores will be released, and they are very, very viable. They can go and they can mate among one another with the other spores. They can form a diploid yeast, and then again asexual reproduction can begin. And in this condition, this yeast can these spores of these yeast can be viable for like hundreds, if not thousands of years, right? So they are very, very viable and sustainable even after a long period of time. So that is why sometimes it chooses to reproduce sexually under unfavorable conditions, right? So moving on to question number 8. The proteins that are targeted to nucleus have a signal sequence that has A, sequences rich in positively charged amino acids, B, sequences rich in negatively charged amino acids, or C, sequences rich in uncharged amino acids, or D, sequences rich in aromatic amino acids. So this is also something that has superficially been discussed in the week's lectures, right? Uh, so there was some discussion about, this is going to be a hint. There was some discussion about some FG, dominant uh, signals that are present in this NLS for some cargo to be imported inside. And some other amino acids were also talked about that are generally present in the NLS sequence, right? So which one were those? Okay, so coming to the answer for this one, the correct answer is sequences rich in positively charged amino acids, right? So these usually have lysine and arginine rich, sequences. Also, let us go through these statements that are written. A nuclear localization signal or sequence, that is NLS, is an amino acid sequence that tags a protein for its import into the cell nucleus by nuclear transport, right? Typically, this sequence consists of one or more short sequences of the positively charged lysines or arginines exposed on protein surface. Also, different nuclear localized proteins may share the same NLS, right? Also, it is not important that it will be presented towards the N terminal or the C terminal or in between the protein. It could essentially, ideally, it could be present anywhere, right? Wherever the important can actually recognize it, right? So in a secondary structure, it should be recognizable or accessible to the importer, but it can be present at any place in the protein that has to be targeted inside. So coming to question number 9, the question is the proteins that are targeted to nucleus have a signal sequence that has the heterochromatin region is, just a moment. Uh, I Okay, I have not changed the question, but I have successfully changed the answers. Let me just go and make this quick change, right? So that I don't give away the answers. Yeah. If somebody's trying to say something, just give me a moment. I don't know why this I've done. Hold on, guys. I'm not sharing my screen currently. Let me just switch this question so that you guys can go through this one also and answer it, right? It was actually pick the correct statement. Okay, let me get back to this. Yeah. So I'll just share my screen again. Right, guys? Just a moment. So this was the last one that we had discussed. And accidentally, I had put the same question in the next one also. But the question that I wanted to actually state was, pick the correct statement regarding nuclear organization, right? So which one of these is the correct statement? Option A is heterochromatin region is darkly stained and transcriptionally active. B is euchromatin region is lightly stained and transcriptionally active. C is histone deacetylation by SIR2 causes euchromatin formation. Or D, that is all of the above. right? So which one out of these is the correct statement? Okay, so we'll go through the answer, which is B, that is euchromatin is a region in the nucleus which is lightly stained and it is also transcriptionally active. Now let us look at the illustration that I have, the image and the illustration, and then we'll discuss why this is, right? So for example, this is an electron micrograph image on your left that you can see, right? So over here, this is the nucleus, right? The cell is there, but we have zoomed it in to a nucleus, right? And in this nucleus, one, you can see a very, very dense dark colored region. Now, this dense dark colored region is nucleolus, right? It is a nuclear body within the nuclear body, and this is where some rRNA synthesis and some other processes that those take place, right? Outside of the nucleolus also, however, you can see that there are some regions that are dark in color and some others that are light, that are gray or white in color, right? So these dark black colored regions, right, they are basically representing heterochromatin, right? And the lightly stained gray or white colored regions are the euchromatin, right? So why are these heterochromatin very dark? Because these are the regions wherein the DNA is very, very tightly packed, right? So these are regions wherein the chromatin or the DNA is very tightly packed. So imagine that you have a white colored thread, right? It is a very, very long thread. But if you really, keep on rolling that thread over and make it more and more and more and more concise, right? So if I keep something on a black background, if I keep that white thread versus a very, very condensed ball of that white thread, you will see that if I take an image on that dark background, this condensed ball will look a little darker than this plain thread, right? So that is what heterochromatin is, right? It is both of them are chromatin, right? Euchromatin and heterochromatin are not different kinds of chromatin. They are the same chromatin that have different kinds of folding, right? That in heterochromatin, the chromatin length that is present is very, very tightly packed. While in euchromatin, that is lightly stained, the chromatin is more loosely packed, right? So if it is not put together very densely, it will stain a little lighter, right? So that is the concept behind this one. And the same thing you can see over here also, wherein a nucleus is shown. In the middle is nucleolus, right? It has been depicted with a different color over here. And then there are dark staining regions that are heterochromatin, which are labeled dark blue over here, right? All the other light staining regions are actually the euchromatin, right? And then we see that there is inner nuclear membrane, outer nuclear membrane, there is nuclear pore complex that is present, right? And together, these inner and outer nuclear membranes are called the nuclear envelope. And just below these, the inner nuclear membrane is a set of proteins which make up the nuclear lamina. Then they are also showing the centrosomes, the microtubules that are emerging out of it. Then the DNA that is associated with proteins or chromatins, plus many RNA and protein molecules are also present within this nucleus, right? And then there are other structures that are represented in this particular image, right? Now we have discussed what heterochromatin and euchromatin is, right? Now let us see which one of these will be more transcriptionally active. So first recall what transcription is, right? So what is transcription? Transcription is basically the relaying or the transfer of genetic information from a gene or a portion in the DNA to RNA, right? So that eventually this RNA will translate this information and form a protein, right? So this gene will transfer some information to RNA, and this process will be known as transcription, right? So for this, the gene has to be available, right? So this gene that is present in the DNA, which is present as chromatin that is complexed with some other proteins, histones and non-histone proteins, it is present inside the nucleus, right? Now, if the histones and the non-histone proteins are present and they are very densely packed and they are very densely packing this chromatin, that is the DNA component, right? Sorry, the DNA component in the chromatin. Then what will happen? The free DNA will not be available for transcription to actually occur, right? Because some portion of free DNA should be there so that the transcription machinery, which is also a very heavy kind of protein complex, will come and sit on that portion of DNA, and then the length of the DNA should be available so that transcription can actually occur, right? But in case of heterochromatin, what happens, since the DNA is so densely packed? How will it be densely packed? Because there will be a lot of proteins that will be present, right? When so much complexity is present, then there will be very little or no DNA that is freely available for transcription to occur, right? So heterochromatin regions, which are densely packed chromatin regions, are transcriptionally repressed, right? But euchromatin regions, which are uh, you know uh, wherein DNA is a little more available, are transcriptionally active regions. Okay, so this is how we can, by seeing just this nucleus, we can say that, okay, we think that in this particular region, there will be more transcription that might be occurring than this particular region. Okay, so I hope that is clear. We'll move on to the final question from the previous year's assignment, that is, the cytoskeleton supporting the nuclear membrane is made up of which one of these? Tubulin filament, myosin filament, lamin filament, or none of the above? So it is a simple question, I think. Um, we have gone through this particular cytoskeleton that supports the nuclear membrane multiple times in earlier lectures as well, right? So the answer for this one is lamin filament. Okay. So in the previous representation also we saw nuclear lamina, right? So why is it called nuclear lamina? Because inside this nuclear envelope, that is a complex of inner and outer nuclear membranes, there is a continuous, protein complex that is present, which is known as lamins, which over here is represented in this green, right? Now, what these lamins do, they support this nuclear envelope, right? So that is their key function. They are just absent wherever the nuclear pore complex is present. Okay. So that is it for the first part of this lecture, wherein we have actually discussed the previous year question for week 5 lecture, right? Now, just give me a moment. Let me just come back to the main screen. Um, yes, guys, just give me a couple of minutes. There was one slight modification I wanted to make to the other PPT, right? There are some credit that I have missed over there. I don't want to present it without that credit. So just give me a couple of minutes and allow me to have water and add that credit. And we can begin with the second part of this lecture, wherein we will go in depth for a few concepts that might not have been discussed in our regular session, right? Because it is a very, very concise session. So it is not always possible for ma'am as well to go into the depth of everything that she is, trying to include and impart the knowledge of. So the things that are left, we try to cover in these extra sessions, right? So that is what the part 2 for the session is going to be about. As I requested, just give me 2 minutes. I'm just going to add the credit and we will begin with the second part of this lecture. So, yeah, yes. So, yes, I'm done with it. I'll just share my screen. Um, okay. We'll discuss some extra concepts, right, that are related to week 5 lectures for this course, that is cell biology, right? For this particular lecture, most of the, things that I have picked up are from these two books that I have used earlier. One is Albert's, that is Molecular Biology of the Cell, the sixth edition of this book, that is written by Alberts et al. And the eighth edition of this other book, that is Molecular Cell Biology by Lordish et al. Right. Also, there are some internet resources and research articles which have been, mentioned wherever they have been used, like I did in my PYQ session, right? So having said that, we can begin with the session. Let me give you a brief overview of what we'll be discussing. So I have, not gone into the depths of nuclear localization in this particular session. I, ran out of time while preparing this session itself. So nuclear localization is something that we can add on to the next week's extra concepts, right? So over here, we have just discussed about meiosis, right? Some other key regulations that occur in meiosis, some key characteristic features that happen when we talk about meiosis, right? How it is different from other phases, and how there are some, special kinds of proteins that are involved in, driving those key differences. So that is what we have discussed in this particular session. As I said, nuclear localization is something we are going to add on in the next week's extra concept session, right? So I did want to speak about it as well, but I ran out of time while preparing the session. Okay. So if we look at an overview of meiosis, like we have already, you might have gone through during week five lectures, right? So it is basically a haploid diploid cycle of sexual reproduction, right? So this could be one of the statements that could be said for meiosis, right? For example, if we take one particular chromosome, so over here for simplicity, the cycle is shown for only one chromosome or chromosome pair, right? In this chromosome pair, we say that there is one chromosome that is coming from the mother and the other that is coming from the father, right? So a single chromosome is being talked about. One pair is coming from the mother and the other is from the father. Both of these, as you can see, are diploid right now, right? But if and when they undergo meiosis, they form haploid, they come into a haploid condition or haploid ploidy, right? So single end ploidy, wherein the egg will only have one copy of this maternal chromosome, and the sperm will have only one copy of this paternal chromosome, right? Why does this happen in the first place? It could have, made another set of diploid chromosomes only, right? That is what happens in meiosis, in mitosis. So why at all do we need this haploid status to be achieved, right? So why we need this is for recombination to occur, right? So for recombination to occur, what do we mean? That in higher organisms and in mammals, including ourselves, what happens is that the progeny that is formed is not identical like any one of its parents, right? So it is not just look like and has the phenotype and genotype only of its mother or only of its father, right? It is like a mix of the two, right? So that is what recombination is. So nobody is identical. The progeny is never identical to any one of the parents, right? This is achieved through sexual reproduction and through sexual fusion and fertilization, right? For this, for us to receive or to come down to one particular zygote or embryo that has a diploid ploidy, right? A 2N ploidy, we need that only one N comes from the mother and one N comes from the father. So for this special condition, so that we can obtain a diploid zygote and not a tetraploid zygote, right? Because if that was the case, if my mother was diploid, I would be tetraploid, and then my progeny will be maybe octoploid, right? So that is not the case, right? Within a species, you have to maintain the ploidy, right? Only then can you consider it as a same species. Right. So for this diploid status to be achieved by the zygote, it was important that only one N comes from the mother and one N comes from the father. Right. It could also have been maintained if we had just obtained 2N from mother and nothing from the father. But then would this zygote not entirely look like its mother? Right. So that such conditions don't arise and recombination does take place, and there's always some kind of betterment that might come along with it. Does this particular process of haploid generation of the gametes occurs, right? So this haploid diploid cycle of sexual reproduction is necessary to occur, and thus we have meiosis in the first place, right? The second thing that I mentioned while we were talking about this is genetic recombination, right? So genetic recombination is very, very important. If we just have meiosis by itself after sexual reproduction, we are anyway going to, we are anyway going to uh, be a recombinant that is produced from our mother and father, right? But despite this, since our mother is just giving one particular chromosome, our father is just giving one particular chromosome, right? Sometimes what happens in some cases, maybe one of those chromosomes is expressing, right? Uh, or there are actually a n number of situations that could occur. We'll go through them one by one. So what could happen is that, for example, if I go back over here, my mother has given me this particular chromosome and my father has given me this particular chromosome. Right now, I am a diploid individual with this set of chromosomes, and due to this, I am a recombinant, right? So, I anyway have different kinds of um a different kind of personality than my mother or my father, because some kind of genes are acting from my mother's chromosome, some kinds of genes are acting from my father's chromosome, some might be clubbed together and might be acting synergistically, antagonistically, we don't know, right? So but I have a very different personality from my mother or my father, right, genetically speaking. So why do we need recombination within these haploid chromosomes also? Why is that? Is Okay, I can't follow my pointer, just a moment. Why is that? Because in case of my mother, right, my mother had these two chromosomes. Out of this, one would have come from her mother and one would have come from her father, right? Similarly, for my father, one would have come from his mother and one would have come from his father, right? So if I just receive the one that has come from the father of my mother, right, and that has come from the father of my father, right? So wouldn't the resultant me uh, just resemble a little of my parents and both my maternal and paternal grandfather, right? But I will not have any resemblance to my maternal grandmother or my paternal grandmother, right? But is that the case? No, that is not the case. Why is that not the case? Because before my mother is passing on one of these two chromosomes, what is happening within the set of chromosomes, right? This is the set of chromosomes that my mother had. Within the set of chromosomes, there is some kind of recombination that is occurring, right? So please forgive my drawing over here because with the mouse. This is the best that I can do. There's some kind of a recombination that is occurring. And this was suppose my mother's mother chromosome, and this was my mother's father's chromosome, right? The plain one. So now some kind of a recombination does occur between these chromosomes before this particular gamete is formed. Uh, Before this particular gamete is formed, right? So after this, what will I receive? For example, this is one chromosome and this is suppose the other chromosome, right? Now this chromosome was my mother's mother chromosome and this one was my mother's father's chromosome, right? So maybe I am receiving this chromosome over here, right? But even if I'm receiving that chromosome, it does have some kind of recombination that it went with my mother's chromosome, right? So now it has the properties of both of my maternal grandparents, right? My maternal grandmother and my maternal grandfather, right? Thus, recombination occurred while this particular gamete was also being formed. Well, this gamete was being formed. Similar recombination also occurred when this particular gamete was being formed, right? After all this recombination has already occurred, there will be this particular gamete will be that will be formed, which will give me the identity of not just my parents but their parents as well, right? So it will be a mixture of these identities that I have, right? So I hope I have not complicated it very much. I hope you understand what I meant to say. Like, if not, I will try to repeat it and I will try to make it a little clearer. So, if that is the case, you can just tell me. If not, we will discuss this slide for which I have actually explained all this, right? So, what I was saying is genetic recombination occurs during meiosis, right? So, as I said, my maternal chromosome is not just one side, one of my parents of my mother's chromosomes, right? It will be a recombinant chromosome, and my paternal chromosome will also be a recombinant chromosome, right? After that, uh, for example, this is one person's chromosome that we are talking about, right? Over here, this both of these are my mother's chromosomes, my mother's chromosome and my mother's father's chromosome, right? There will be some kind of crossing over that happens. And after that, these are the haploid gametes that we receive, right? So, for this also, there is some places wherein the crossing over will happen more than the other regions, right? We will read about that later. Also, over here, it is mentioned that the greater the distance between the two loci on a single chromosome, the greater is the chance that they will be separated by crossing over occurring at a site between them. Okay. If two genes are thus reassorted to be in X% of gametes, they are said to be separated on a chromosome by a genetic map distance of X map units or X centimorgans. So, map units or centimorgans is basically an arbitrary unit. It is named after this person, Morgan, right? Who is one of the famous geneticists, right, who had studied these recombinations in flies. Over here, what they are saying is that the greater the distance between two loci. So, for example, this is one loci and this is the other loci. The distance is huge, right? So, there is more chance that they will be separated, and they are indeed separated, right? Now, this loci is different and this loci is different, right? They are separated. Sorry, let me explain through this. This is one loci, this is another loci. So, since they are so far apart, you can see that they have been separated in these two cases, right? But if the loci are very close by, right? For example, this is one loci I am talking about, and this is another loci. So, loci is basically a position on the chromosome. Any one spot on the chromosome is known as a loci. Okay? So, if these are the two loci, on the other hand, that I'm talking about, which are very, very close by, right? These two, we see that they are together on this one also, and they are together on this one also, right? So, the greater the difference, distance between two loci, the higher will be the chances. Everything is a chance event. It might occur that the crossover takes between these two also, right? That might occur. But since they are close by, it will occur less frequently than crossing over taking place between this and this loci. Okay, so we actually talk about it in more emphasis when we talk about genetics. Since we are studying cell biology for now, we will not go into the genetic details of this thing, right? But this is a very, very interesting topic, and there are some numericals also that we solve in genetics related to this. So for that, you can, refer any of the genetic books also. Plug and Cummings is one, human molecular genetics is another. So that is up to you. Uh, okay, so we'll just come back to cell biology for now. And we will talk about meiosis, right? So for meiosis to occur, like we have seen that what is meiosis, but we want to know that for it to occur, there will be some kind of signal, right? So what is this kind of signal, or what is this kind of cue, right? So we know that extracellular and intracellular cues are regulating the germ cell formation, right? So it could either come from outside the cell or within the cell, right? So extracellular signals induce a transcriptional program that produces meiosis-specific cell cycle factors that bring about the unusual meiotic cell divisions. Okay, this modification of the cell goes hand in hand with a developmental program that induces the features characteristic of the gametes. Okay, so I hope these statements are self explanatory, right? So we are just saying that extracellular signals could basically switch on some transcriptional machinery, which could, recruit factors that are necessary for meiosis to occur. Okay, and when this will happen, then simultaneous development of machinery that will actually prepare these germ cells will also start. Okay, also for mammals, for example, these factors or these cues have not really been understood very well. So we don't know, in case of humans or in case of higher order animals, we don't really know what is basically driving them to actually form or undergo meiosis and form these gametes, right? We don't know the exact cues, but there are, since there has been extensive research that is done, and there is extensive research that is going on also, we have some answers. Okay, so we know at least one of the extracellular signals that induces entry into meiosis in mammals, right? Which is this extracellular signal? It is known as retinoic acid. So retinoic acid is a ligand, right? What will it do? It is a signaling molecule that, by binding to the transcription factor retinoic acid receptor, it will function in many different developmental processes, right? So once it initiates these developmental processes, it will also cue these processes to undergo meiosis. Okay, the cellular targets of this particular hormone, that is retinoic acid, and how it functions to specify the meiotic fate remain to be discovered, right? So from this extensive, really long and really many studies, we have definitely determined that this is one of the extracellular signals that is involved in, regulation of the cell's fate into meiotic division, right? But we don't really know how this is happening, what alterations it is bringing. So maybe 10 years down the lane, a couple of decades down the lane, we do understand this better. Okay, but there is indeed one organism that we have discussed about earlier also, regarding which we do have knowledge about these cues that can actually regulate germ cell formation. Which is this animal? This animal is the budding yeast, right? Or cerevisiae, right? Saccharomyces cerevisiae. So, as we have discussed before, sorry, this organism can undergo asexual as well as sexual reproduction, right? So, when there is mating between haploid cells of opposite mating type, so consider one as a female and the other as a male, right? So that is an opposite mating type. right? Then diploid cells are formed, right? Then through asexual reproduction or vegetative growth of diploid cells, it keeps occurring, right? And buds are formed. Now these buds will continuously be forming, like now this bud is formed. Now these two cells are there. Now each cell will form more buds, another bud, right? Now four cells are formed. Each of these will form more buds. Now, it will keep doubling, right? Till the conditions are favorable. But if the conditions are not favorable, again, like we spoke about earlier, then starvation will occur and it will cause the ascus formation and meiosis, right? So, what is this ascus formation? In this, there will be a formation of an outer covering, which is very, very resistant to most of the stresses that are present externally in nature. And within this covering, there will be four haploid ascospores that will be formed, right? So from that one particular yeast that has formed the ascus around it, it will have gone, DNA duplication. It will become tetraploid. From that, four different ascus spores will be formed. And when the conditions are favorable again, this ascus will rupture and spores will germinate, right? After this, there could occur vegetative growth of these haploid cells, right? So even haploid cells could undergo vegetative growth. They could, increase in number by it, and then mating will occur again. And now again, diploid cells will be formed, right? So that is the life cycle of a budding yeast or Saccharomyces cerevisiae. Now, how or what is the cue for this germination to occur, right? So we know that starvation is causing it. That is one cue, right? There are some cues that we have already talked about also earlier, right? For example, IME1, IME2 checkpoints, which will act as transcription factors that is going to, regulate these meiosis process, right? This meiosis process. So let us talk about it in a little more detail. Right. So the decision to enter the meiotic divisions is made in G1 phase. Okay. The depletion of nitrogen and carbon sources induces diploid cell to undergo meiosis instead of mitosis. So these are the nutrient deficiencies that we have been talking about. And this yields haploid spores. Okay. After this, what will happen is during the meiotic divisions, the budding is repressed. So while meiotic division is taking place, there will be a repression of the budding that was going on, right? Because right now all the energy needs to be, put on and kind of navigated for this meiosis to occur correctly, right? So four haploid cells have to be formed. Right now, the time for meiosis, for mitosis or budding to occur is not there, right? The pre-meiotic S phase and the two meiotic divisions that occur within the confines of the parent cell. Okay, the spore walls are then produced around the four meiotic products. So first the parent cell is present, its wall is already present, right? It will undergo these two meiotic divisions. The four kinds of spores will form and then the walls around these four meiotic products will also form, right? The nutritional starvation represses expression of G1S phase cyclins, thereby it inhibits budding. Right. So we know that how is budding inhibited? Because G1SP cyclins are inhibited. Why? Due to nutritional starvation. But now a very, very, keen question arises that if the G1SP cyclins are repressed, then how does the DNA replication for meiosis take place? Right? So it is not that we have to go through the S phase only for mitosis. We also have to go through the S phase for meiosis, right? For that also we need the duplication of DNA to occur, right? So if these cyclins are not there, how will this duplication take place? That is the key question. For this, there had been experiments that were performed, and now we know that the sporulation specific protein kinase, that is IME2, takes over the role of G1S phase CDKs in promoting DNA replication. Okay, so this is a protein kinase that is specific only for sporulation. Okay, the IME2, what does it do? It promotes phosphorylation of the APCC specificity factor CDH1, which inactivated to the S phase and M phase cyclins can so that the S phase and M phase cyclins can actually accumulate. After this, the phosphorylation of the transcription factors is done to induce expression of genes whose products are required for S phase, including the DNA polymerases and S phase cyclins. Then phosphorylation of the S-phase CDK inhibitor SIC1 is done to leading to release of active S-phase CDKs and onset of premeiotic DNA replication. Right, so there are these three kinds of, this is a kinase, right? What is the property of a kinase? It will phosphorylate or it will add a phosphate group, right? So it will add the phosphate group to APC to basically inactivate APC and to accumulate S phase and M phase cyclins. Then it is going to add a phosphate to transcription factor so that S phase polymerases and S phase cyclins are available now, and it will also phosphorylate the S phase CDK inhibitor SIC, which will further lead to you know uh, activation release of active CDKs, S-phase CDKs, right? So because some kinds of phosphorylations are activating, some are inactivating or inhibiting. So this one is an inhibiting phosphorylation, wherein this since this inhibitor is phosphorylated, now active S-phase CDKs can actually be released, right? And this can help in DNA replication. So once that has been done, and the meiosis actually starts, we see that in meiotic prophase, there are duplicated homologous pairs of homologous chromosomes that pair up, right? So this is another characteristic feature of meiosis, right? Because when we talk about mitosis, the homologous chromosomes don't pair up, right? They remain separated and they move towards opposite spindle poles, right? But during meiosis, duplicated homologs pair. Right. So during meiosis 1, it is crucial that the homologs recognize each other and associate physically in order for maternal and paternal homologs to be bi-oriented on the first meiotic spindle, right? So why is this important? Uh, what is the first criteria for this? That first, they should be able to recognize their homologous chromosomes, right? That is not key or that is not important in mitosis, right? But over here, that is very important because you have to go and bind physically to that particular homologous chromosome, right? Then meiotic prophase or prophase 1 can take hours, right, in yeast, and it can take days in mice, and it can take weeks in higher plants. And for eggs, for example, in eggs, we know that it can even take years, right? For example, in humans, there is this menstrual cycle that occurs wherein the eggs are arrested in metaphase 1, I think. If I am not getting it, wrong, then metaphase 1 of meiosis 1, right? So they are arrested in meiosis 1 or metaphase 2 it is. Okay, so I will check my facts actually. So I don't want to state anything incorrectly. But meiosis can actually take years also to occur, right? One of the examples is the egg development in humans, right? And prophase itself can take like hours, days, or even weeks, right? So having said that, uh, we see how this pairing of the duplicated homologs actually occurs in the meiotic prophase, right? So like their mitotic counterparts, duplicated meiotic prophase chromosomes first appear as long thread-like structures. Like that is how they will earlier appear. Like for example, in prophase 1's first phase, that is leptotene, they will still appear like thread-like structures, like we talked about in another representation that we saw of that synaptonymal complex assembly and disassembly. We saw that it was still looking a lot like or very intermediate between interphase and the packetine stage, for example, right? Also, it is during early prophase 1 that the homologues actually begin to associate along their length in a process called pairing. Okay, which in some organisms at least begins with interaction between complementary DNA sequences called pairing sites in the two homologous chromosomes, right? So if we are saying that two chromosomes are homologues of one another, means that they have very similar properties. So there will be some complementary DNA sequence sites that will be present, right? Now, these will be the pairing sites uh, among which there is actually physical binding that will actually occur, right? Okay. So, as the prophase progresses, the homologues become more closely juxtaposed. So they are brought together, right, forming a four chromatid structure called bivalent. Right. So what is a bivalent? It is a four chromatid structure. In one particular chromosome, there are two chromatids, right? They are known as sister chromatids. When we talk about two homologous chromosomes, there will be four chromatids. Two chromatids will be sister chromatids among themselves, and the others, they will be non-sister chromatids to the other two chromatids of the other chromosome, right? But all these four chromatids will be brought together, or these two chromosomes will be brought together, and the structure will be known as bivalent, right? So remember this. This is another term that you might be asked a question on, right? Okay. So after this bivalent is actually formed, the homolog pairs and are then locked together by homologous recombination, and the DNA double-strand breaks are formed at several locations at the sister chromatid, resulting in large number of DNA recombination events between the homologs. Right. So over here, for example, you can see these small arrows. Right, they represent double-strand break, and you can see that there are a lot of double-strand breaks that will occur along these non-sister chromatids, right? Or along these homologous chromosomes. And once this actually happens, there will be a lot of sites wherein crossing over or this DNA recombination can actually occur, right? But some of these events lead to reciprocal DNA exchanges called crossovers, where the DNA of all of a chromatid crosses over to become continuous with the DNA of homologous chromosome. We have seen what crossing over looks like, and I hope this statement makes sense with context to that, right? Also, having said that, we are saying that there are multiple kinds of DNA breaks, that double-stranded breaks that are occurring, right? And there are multiple possibilities of DNA recombination. However, not all these recombinations will actually occur, right? So the crossing over or the homologous recombination will actually take place at around one or two or maybe three sites, right? Not all of these sites will have homologous recombination between them. That will occur, right? That is how it is regulated. Okay. So homologous pairings also culminates in the formation of a synaptonymal complex. Okay, so what is a synaptonymal complex? We will talk about it. The large protein machine is called, which is called a recombination complex, assembles on the double strand break, right? Wherever there is a double strand break, there is this large protein complex, which is known as recombination complex, that will come and sit on it and in a chromatid, and it will bind the matching DNA sequences in the nearby homolog pair and help reel in its partner, right? So it will kind of try to identify its homologous partner. It will try to identify similar sequences wherein the breaks have occurred, and then it will try to bring it close by, right? This so-called presynaptic alignment of the homologs is followed by synapses, right? So during this presynaptic alignment, for example, let me give you an idea. These homologous chromosomes are about 400 nanometer apart, right? During this thing that is still occurring. After that, synapses occurs, right? After synapses will occur, the actual core of the homolog becomes tightly linked to the actual core of the of its partner by a closely packed array of transverse filaments to create a synaptonymal complex, which bridges the gap, which is now only 100 nanometer between the homologues. So before the synaptonymal complex had formed, right, after which the gap is only 100 nanometer, before it had formed, the homologues had still come by close together and the gap was around 400 nanometer, right? After that, these axial cores, these, right? So there is this one set of homologous chromosome. This is another set of homologous chromosome. In both, you can see that there are two sets of sister chromatids, right? In both of these, there are cohesion rings that are keeping these sister chromatids together, right? And there are these axial cores of both these homologues that are different, right? And they are brought together by transverse filaments that are present close to it, which is what was being talked about. Okay. So, coming back to this representation that we had already seen earlier, as we talked about, there are these four chromatids that are present, right? Now, two of these, 3 and 4, are sister chromatids, 1 and 2 are sister chromatids. Now, these, along with them, have an axial core complex, right? This slightly green colored complex is the axial core complex, right? That is forming uh, uh, around, in complex with one set of sister chromatids, right? And between them is this transverse filaments, right? These transverse filaments are these, right? So once these axial core complex and these transverse filaments come and tightly packed together, the synaptonymal complex will be formed, right? The assembly of this complex starts in zygotine and its disassembly begins in diploidine. Okay, in packetine, they are very tightly assembled. Okay, so talking about the synaptonymal complex. The prophase basically starts with leptotine when homologues condense and a pair of genetic recombination begins. At zygotine, the synaptonymal complex begins to assemble at sites where the homologues are closely associated and the recombination events are occurring, right? After this, at packetine, what will happen? The assembly process is complete and homologues are synapsed along the entire length, right? Along the entire length, like from here to here, the homologue will be synapsed. The packetine stage can persist for days or longer until desynapsis begins at diploidine stage, which with the disassembly of the synaptonymal complexes and the concomitant condensation and shortening of the chromosomes, right? It is only at this stage, after the complexes have disassembled, that the individual crossover events between non-sister chromatids can actually be seen. Okay, during this synaptonymal complex, since both the homologues are entirely fused together, right? You will not be able to make out where the recombination event has taken place. But after this synaptonymal complex has disassembled, these homologous chromosomes will be still attached at the point of recombination. So after this only it will become apparent, right? That the individual crossover events between non-sister chromatids can be seen as interhomologue connections called chiasmata, right? Chiasmata is plural for chiasm. Uh, Chiasma is actually plural for chiasmata, which now play a crucial part in holding the compact homologues together. Okay. So over here, for example, after the synaptonymal disassembly has taken place, now we can see, like, if you can look at this micrograph image and this image that has been drawn looking at it so that it is easier for you to understand. If you look at this micrograph image, okay, just a moment, guys, let me just attach a charger. So I have attached a charger. What I was saying was that if you can imagine that after the disassembly of synaptonemal complex has occurred, this is what the bivalent looks like, right? You can actually see that these are the sites that at which the two homologues are attached. So these are probably the three sites wherein crossing over has actually occurred, right? But if we had seen it at the packeting stage, what we would see was an electron micrograph image, which kind of looked like this. Okay, these are also very far spaced only because this is the best I am being able to draw. Like, imagine me having drawn four chromatids and they look like this. Okay. So they are so close by together that you will not be able to make the difference, right? That where the crossover has occurred, if you understand, right? Just a moment again. So, having understood that as well, we will see how homologue segregation depends on several unique features that are unique to meiosis 1. Okay. So, a fundamental difference that exists between meiosis 1 and mitosis, or even meiosis 2, which resembles mitosis largely, is that meiosis 1 homologues, rather than sister chromatids, separate and then they segregate, right? This difference depends on three features of meiosis 1 that distinguish it from mitosis. Okay, so what are these three features? Let us go through them. The first one is that both the sister kinetochores in a homologue must attach stably to the same spindle pole. Okay, so in mitosis, what we talk about is that the different sister chromatids should attach to different spindle pores so that they are, segregated when we are separating this complex, right? But over here, we want that the sister kinetochores should attach to the same spindle pole. Sorry. Right. For this, the two sister kinetochores are fused into a single microtubule binding unit that attaches to just one pole. Right. So this will make it very easy if they are not separate units, but they are fused as one unit so that the tubule will come and attach to that one unit, right? So they will attach to the same microtubule. Fusion of sister kinetochores is achieved by a complex of proteins that is localized at the kinetochores in meiosis 1. Although we don't really know what this complex protein is, right? What this protein complex is, right? They are removed from kinetochores after meiosis 1 so that in meiosis 2, the sister chromatids pair can be bioriented. Bioriented means it can attach to different poles and then they can, be segregated on the spindle as they are in mitosis, right? So over here, you can see that instead of the two different sister chromatids in red and two different sister chromatids in gray being attached to opposite poles, they are attached to the same pole, right? So the gray ones are coming and segregating to one pole, the red ones are segregating to the other pole, right? So that is how this is happening. Sorry. The second one is the crossover generates a strong physical linkage between the homologues, right? So what does that mean? That see over here, we know that there is chiasmata that are present, right? So chiasmata are physical crosses that are present between different homologues, right? So, since these physical linkages are present, they allow their bioorientation at the equator of the spindle, much like cohesion between sister chromatids. It's important for their bioorientation in the mitosis, right? Also, crossovers hold homologue pairs together only because the arms of the sister chromatids are connected by the sister chromatid cohesion, right? Okay. The third one, which is the more important one, is that the cohesion is removed in anaphase 1 only from the chromosome arms and not from the regions near the centromeres. So this is something that was discussed while in your week 5 lectures. You were being taught meiosis 1 or the concept of meiosis 1. This is one thing that had been covered over there. There will be one additional thing that we will see in this, right? So, what are we saying? That when the cohesion is also removed, right, as happens during mitosis, also during meiosis 2, also it will happen that the cohesion rings are removed, right, through the action of separase. So, when this is removed, in anaphase 1, it is only removed from these arms of chromosome. It is not removed from the centromere region, right? Uh, near the from the and it is not removed from regions near centrosome where the kinetochores are located. Okay, the loss of arm cohesions triggered homolog separation at the onset of anaphase 1. This process depends on APC complex activation, which leads to securrin destruction, separase activation, and cohesin cleavage along the arms, right? But there is a reason why only around the centromere this cohesion is not segregating, right? So the question is, how are cohesins only near the centromeres not removed, right? They are removed from the arms. Over there also, if the separase activation had been inhibited, then along the arms also it would not have been possible. But over there it has been separated. Only near the centromeres it has not been separated. Now, why is this so, right? So this is so because cohesins near the centromeres are protected from separates, right? How are they protected? And specifically in meiosis 1, they are protected by a kinetochore associated protein, and the name of this protein is shugoshin, right? So I think it is a Japanese word which means guardian protein, some kind of a guardian, right? So it is guarding this disassembly, right? Shugoshin acts by recruiting a protein phosphatase that removes phosphates from centromeric cohesins. Then cohesion phosphorylation is normally required for separase to cleave cohesin, right? But now, since this phosphorylation has been removed, the removal of this phosphorylation near centromere prevents cohesion cleavage, right? Cohesin cleavage, sorry. The sister chromatids pairs therefore remain linked through meiosis 1. And after meiosis 1, shugoshin is inactivated, due to which the phosphorylation again can occur. And in meiosis 2, now they can actually, the cohesion can be removed by the activity of separase. And now the sister chromatids can also be moved apart, right? And another thing that was talked about in the week's lectures is that crossing over is highly regulated. Now, yes, indeed, it is highly regulated. There are not only the number, but the location of double strand breaks along the chromosome is also controlled, right? As is the likelihood that a break will be converted into a crossover. So we saw that there are multiple breaks, multiple double-stranded breaks that can occur, but not all of them will actually be converted into a crossover, right? On an average, the result of this regulation is that each pair of human homologs is linked by about 2 or 3 crossovers. So this is our limit, right? Most of our chromosomes undergo 2 or 3 crossovers per meiotic division. Okay. Now, double strand breaks occur in meiosis 1, and they can be located almost anywhere along the chromosome, but they are not distributed uniformly. They cluster at hotspots where the DNA is accessible and occur only rarely in cold spots such as heterochromatin regions and around centromeres and telomeres. So heterochromatin, I told you, is a region wherein DNA is not very accessible, right? So it is one of the cold spots. Also, centromere is another region wherein there is this cross or physical connection that is occurring. So over there also it doesn't happen. And at the edges near telomeres also, usually double strand breaks don't occur, right? They usually occur at more DNA accessible regions, which are known as hotspots, right? Then there are two kinds of regulations that influence the location and number of crossovers that form, neither of which is well understood. Okay, but both operate before the synaptonemal complex assembles. So we know that before these two homologs are coming together, this has been fated, or this has been decided where the crossover will be formed and how many crossovers will be formed, right? One of these uh, functions, they ensure that at least one crossover forms between the members of each homolog pair, as it is necessary for normal homolog segregation in meiosis 1. The other regulatory mechanism is known as crossover interference, which means that presence of one crossover event inhibits another from forming close by, perhaps by locally deleting proteins required for converting a double strand DNA break into a stable crossover. Okay, so this also I think we have gone over earlier. The crossover interference, what it will be, the two crossovers can't occur like next to one another, right? If there is one crossover that is occurring, it will inhibit physically the other crossover to occur within a certain distance, right? After a certain distance, it might be able to occur. Okay. So, which also tells us that maybe in smaller chromosomes, the number of crossovers might be lesser than in the longer or larger chromosomes, right? So, as we have discussed, there could always occur errors in meiosis, like the errors that we were discussing due to non-disjunction of chromosomes, for example. Right over here, I have mentioned some examples of these errors and how, some kinds of diseases or some kinds of syndromes arise from these errors, right? One of them is Down syndrome, right? So over here, Down syndrome we have gone through. It means the trisomy of chromosome 21, right? So this means that instead of the normal two copies of chromosome 21 that should be present, now we have three copies of chromosome 21 that are present, right? So this will cause some kind of a defect or deleterious. It will be somehow deleterious to the person or individual who actually has this trisomy 21, right? Then there is another syndrome, which is known as Turner syndrome, right? What happens in Turner syndrome? Now, over here, you can see that in the sex chromosomes, there should always be two sex chromosomes. It could either be two X chromosomes or one X and one Y chromosome. Over here, Turner syndrome, there is one X chromosome, right? Only one single copy of X chromosome is present. So individuals who do have Turner syndrome, what will happen, there will be some kind of infertility issues that they will face. Then they will not be a normal level of feminine also, right? They will have some maleness because some level of the hormone regulation that occurs due to the second X chromosome will not happen in these particular females, etc., right? Then there's a third kind of syndrome that could occur due to it. There are a lot of them. Okay, I have mentioned just four examples over here. This is Klinefelter syndrome. So I have given you a karyotype for this also. Uh, Look at it and see how this one is defective. Okay, so since we are anyway over time, I will tell you that over here, like I said, we should have two copies of sex chromosomes, one X, one Y, or two X's, right? Over here, we have three copies of sex chromosomes, right? Two X and one Y. So since this individual has one Y chromosome, the person will probably be a male. But since the male also has two X chromosomes, it might be a male who has a lot of hormonal defects or a feminine, nature. Again, it could be an individual who would have issues in fertility, etc. Right? It could be there could be a number of problems that could occur, right? You can go and read about it. The final one that I have mentioned over here is Edward syndrome. Right? What is Edward syndrome? Edward syndrome is the trisomy of chromosome 18. Right. Due to this, again, there will be a lot of developmental defects postpartum that will occur. And people who have Edward syndrome again have some classical characteristic features, due to which it is difficult for them to lead a normal life. Right. Then these are the next two slides are something that I have already discussed, but I have nonetheless kept it in this week's lecture for people who had not attended my week two lecture, right? So over here, there is a difference between mitosis and meiosis that has been represented over here. I am not discussing it again over here for more insight on this. You can maybe go and look at the week two practice session that I had taken. Also, I have kept the differences or the key differences between mitosis and meiosis, right? Over here, there are some key differences I would really like to go over again. Right. Since we are a little over time, we will go through them quickly. So we know that in case of mitosis, let me just enlarge this segment. Yeah, so we know in mitosis, one cell division results in two daughter cells. While in meiosis, we know that one cell division, there will be two cell divisions that will occur, right? Meiosis 1 and meiosis 2, and the result will be four daughter cells, right? Now, in mitosis, the chromosome number per nucleus is maintained. So when we start with a diploid organism, we receive two diploid daughter cells. But in meiosis, the chromosome number is halved, right? Because the first division is reductional division, the second is equational division. So 2n results in four haploid progeny, right? Then, during mitosis, there is one pre-mitotic S phase per cycle division, right? So one S phase division occurs. After that, one mitosis will occur. Now, in meiosis, also there is only one pre-meiotic S phase. That is why the chromosome is reduced to half. The ploid is reduced to half, right? Because there is one S phase division, but consequently or subsequently, there are two cell cycle divisions that are taking place, meiosis 1 and meiosis 2, right? Then in a case of mitosis, normally there is no pairing of homologous chromosomes in prophase, but there is a full synapsis of homologous chromosomes in the prophase and meiosis, right? Then there's normally no recombination in prophase in case of mitosis, but at least one recombination between non-sister chromatids occurs in case of meiosis. Also, there is bioorientation of sister kinetochores and loss of cohesion between sister chromatid arms during prophase, but we have gone through this, that during meiosis 1 at least, co-orientation of sister kinetochores occurs in meiosis 1, so they will move to one particular spindle pole. Also, maintenance of cohesion between sister chromatid only around the centromere, not around their arms, will occur during prophase of meiosis 1. Okay. Then centromeres divide at anaphase, but in this meiosis, centromeres will not divide at anaphase 1, but they will divide at anaphase 2. Okay. And mitosis is a conservative process, and the daughter cell's genotypes is identical with the parental genotype, and the cell undergoing mitosis can be diploid or haploid. While in case of meiosis, it promotes variation and among the products of meiosis, so it is non-conservative. And cells undergoing meiosis is diploid or multiple thereof, right? So an haploid cell will not be able to undergo the sexual reproduction division that is known as meiosis. So that brings us to the end of today's discussion as well. I will just stop sharing my screen. I hope what I aimed to discuss was clear. Like I said, I was unable to add the extra concepts regarding the nuclear localization of materials, right? So that we will include in next week's discussion. And that was it from my side for the day. If there are any questions, you are most welcome to answer them. Okay. So if not, I will end the session, and first let me stop the recording.